Hello again and welcome back to the channel. In this lesson we're going to be looking at active transport. It'll be broken up into the following sections. First we'll look at what active transport is, and then we'll talk about why active transport requires energy and in what form that energy takes. We're also going to talk about the main forms of active transport, giving some detailed descriptions of how active transport works in cells. And then finally, we'll talk about how ATP works and what its link is to active transport. So let's have a look. So as usual, the worksheet is linked in the description to the video below. You can download it using the link, and then you can fill it in as we go along, either by printing it out or by doing it online, say for instance on an iPad, which is actually the best way to do it. So let's start trying to fill this in together. So on the first page it says the active processes used by cells to transport substances in and out of them require what in the form of ATP, and they require energy. So they require energy in the form of ATP. And the reason why is because they're moving molecules against their concentration gradient. And we'll talk about what that means in a minute. So the main active processes you need to know are, number one, just straightforward active transport. So this process moves molecules against their concentration gradient. So let's see if we can define active transport. So active transport is the movement of molecules from an area of lower concentration to an area of higher concentration. In essence, that means that the molecules are moving against their concentration gradient. So they move against their concentration gradient. So when we looked at osmosis and diffusion in an earlier video, we talked about how in those processes the molecules moved down their concentration gradient from an area of high concentration to an area of low concentration to try to get to an equilibrium on both sides. In this case, you're doing the opposite. You're moving the molecules from an area of lower concentration to one of higher concentration. Basically, you're concentrating them more in another area. That requires energy to force the molecules to do that. Okay, so at the bottom, you've got a diagram that shows one way in which active transport could work. And as you can see, you've got a green molecule, a protein in the membrane, embedded in the membrane of the cell. And that protein can temporarily bind to a substance and then allow it to pass through the membrane and across either into or out of a cell. And as you can see, that movement is coupled with dephosphorylation of ATP to ADP, and that enables the process to occur. So there are lots of examples of active transport in living organisms. I'll give you one of them. So an example of active transport is the absorption of minerals into root hair cells in the soil in order to use them and to decrease the water potential. So this symbol, what looks a bit like a trident, it's the Greek letter psi. So a bit like a trident, like as in Poseidon, which is the Greek god of the sea. So it lowers the water potential to absorb water. So it enables the roots to absorb water from the soil, because if you artificially, or if you create a situation in which the water potential within the root tissues is decreased, then water is going to be absorbed into the roots by osmosis. So that's one example of active transport. As usual, there's a QR code just there. And that links to another useful YouTube video on this topic. So let's go on to page 2. So on page 2, you've got another example of active transport in cells, what we call endocytosis. So endo, from Greek again, means to take something in. So this is another active process, thus requiring energy, which involves a useful feature of cell membranes. The membrane surrounding organelles and the main cell membrane are made of the same main component, what we call phospholipids, they're a type of fat. Endocytosis is used by cells to bring substances or particles in. Now you can see in the diagram I've provided some examples of endocytosis. You've got phagocytosis, which is bringing in solid particles, or say for instance pathogenic cells, into a cell by making a pocket of their membrane and then forming a vesicle, basically a membrane sac around whatever you're engulfing, and then taking it into the cell. And then in some cases, it could be then fused with something called a lysosome, which contains enzymes which will break down that solid particle. Another example is penocytosis, which is where a cell takes in some of the surrounding liquid around it. It takes in everything that's dissolved in that surrounding liquid, and then it's able to make use of whatever it needs from that surrounding liquid. And then finally, in this diagram, you've got receptor-mediated endocytosis. So that's a bit like penocytosis, except you're specifically picking, or the cell is specifically picking, the molecules that it wants to take in. How does it do that? It uses receptors on the surface, on the membrane, which are complementary in shape. Not the same shape, but complementary in shape to the molecules that it's trying to take in. And so they bind to those molecules, and then the molecules can then be taken in. So if you look at the diagram, the stars are complementary to the receptors. And so only the star-shaped molecules will bind, and only they will then be taken in to the vesicles that are ingested by the cell. So let's write some stuff down with regard to what we've just been talking about. It's always a good idea to write material down because it enables you to retain the material a lot better. So, phagocytosis. This involves cells taking in solid particles in pockets of cell membrane which form vesicles. Okay, so vesicles are basically just membrane bound sacs. Okay, so next, penocytosis. This involves cells taking in samples of surrounding liquid which contain dissolved substances which the cells might want for cellular processes. Okay, so basically you've gone from the first example I gave you, which is phagocytosis, which is taking in a solid particle, or say for instance a cell, into another cell. In penocytosis, you're just taking in a liquid that contains dissolved substances within it, which the cell may or may not need. It'll take what it needs and get rid of whatever it doesn't. Okay, and then finally we have receptor mediated endocytosis, which is a bit more precise. So this involves cells using receptors to take in molecules. Well, actually, let's make that specific: molecules. Molecules from their surroundings, e.g. extracellular tissue or extracellular fluid. This is very specific. It means that the cell can actually pick and choose what molecules it wants to take in, dependent on whether or not those molecules are complementary in shape, as I said earlier, to the receptors on the cell surface membranes. Okay, so when might endocytosis be needed? Endocytosis is required when cells want to take in substances or particles from their surroundings. So let's do some examples. I've already mentioned some of these, but we might as well write them down. So one example is when immune cells engulf pathogens from their surroundings. So immune cells let's sort out some of my horrible handwriting there. Immune cells, white blood cells, white blood cells ingesting pathogens. Or another example might be cells ingesting or taking in substances which they require in order to make other substances. So cells taking in large molecules from their surroundings. Why do I say larger molecules? Basically, if the molecules were very small, they'd simply be able to pass through the membrane directly. However, if they're larger, or if they are, for instance, charged and can't get through the membrane directly, then the cell could use something like endocytosis. Okay, so let's look at the opposite of endocytosis, something called exocytosis, like exit. Okay, so in exocytosis, the particles or molecules are moving out of the cell. It's very, very similar to endocytosis, except the particles or molecules are moving in the opposite direction to what we saw in endocytosis. So if you look at the diagram, that shows that quite nicely. You've got something like a secretory vesicle. So a secretory vesicle is a vesicle that contains a substance that a cell wants to get rid of and pass into the extracellular fluid. So you can see that the membrane-bound vesicle fuses with the membrane of the cell and then eventually lets out the sort of teal or light green colored circular or spherical molecules into the extracellular fluid. Okay, so let's write something down for that. So exocytosis. Exocytosis involves the removal of particles or substances from cells by fusing membrane-bound vesicles with the cell surface membrane. And as such, whatever substances or particles are contained within the vesicles are then let out of the cell into the extracellular fluid. Okay, so ATP. ATP is, in essence, the energy currency used by cells. What do I mean by an energy currency? When you go on holiday, let's say you go to, I don't know, somewhere in mainland Europe, you would need to use euros because they won't accept pounds. And that's because the currency used in the Eurozone is the euro, whereas the currency used in the United Kingdom is the pound. And so they won't accept pounds and we don't accept euros. So you would have to convert your currency into the local currency in order for you to be able to use it. This is very similar here. Energy can be moved around in all sorts of different forms. However, cells can only use energy directly in the form of ATP. So whatever other form of energy comes into the cell, it has to then be converted into ATP. And we normally do that, or our cells normally do that, by using respiration. And so you'll have a bank of ATP within your cells, which can then be used for all sorts of cellular processes, including active transport. So let's find out what ATP actually stands for. ATP is just an acronym. It's a way to shorten a longer name. And so ATP stands for adenosine, that's the A, triphosphate. So that's ATP. A for adenosine, T for tri, and then P for phosphate. Okay, and that simply means that the ATP has 3 phosphate groups attached to it. So that's in its charged form, so to speak. So we can think of ATP as being a bit like a rechargeable battery. Rechargeable batteries are much easier ways to, or more efficient ways to actually power your electronic devices. So when I was much younger, I remember my mother being infuriated whenever I went through vast numbers of batteries for things like remote control cars and whatever else. And actually, it was a godsend when I could get hold of rechargeable, in those days, nickel cadmium batteries. These days, you're more likely to use lithium ion batteries. And they're a lot more cost effective because you can use them, recharge them, use them again, recharge them, and so on for a certain number of cycles. ATP is more or less the same sort of thing, but for cells. And what can happen is you can store energy in ATP, take it out for cellular processes, and then restore more energy back in, and then just continue doing that cycle a certain number of times. So if we look at the diagram, we can see how that works. Constantly, the ATP cycles between ATP and ADP, which is adenosine diphosphate, which contains 2 phosphate groups. So when you make a bond between the 2 phosphate groups in adenosine diphosphate with another phosphate group to make adenosine triphosphate, you trap energy in the bond that you formed. And when you want to get that energy back, you just break that bond and it releases that energy, which can then be used by the cell for whatever process it needs. As usual, there is a QR code which will take you to a relevant video that summarizes everything I've just said about how ATP works. So you can look at that in your own time. So that is it on active processes. You don't need to know quite as much about active processes as you do for the passive processes like diffusion and osmosis. And quite a lot of people seem to struggle with osmosis. So I suggest you look at the video I made earlier about osmosis. Just go to the channel and you can find it. So let's have a quick summary of what we have learned today. So active transport involves moving molecules against their concentration gradient, basically moving molecules from an area where they're in lower concentration and moving them to an area where they would be in a higher concentration. Of course, that means that you need energy. And the reason why you need energy is because you're forcing the molecules to move in a direction that they wouldn't naturally move in. Okay, and then the next bit we talked about what form that energy would be in. So ATP is the form in which energy is used by cells for processes like active transport. You need that energy, as I said, to move the molecules against their concentration gradient. And we also talked about some examples of active transport, including endocytosis, which can then be broken down into things like phagocytosis and penocytosis and receptor mediated endocytosis, all of which are examples of active processes which bring molecules or particles into cells. And then we talked about exocytosis, which is the opposite of endocytosis. It's how cells remove particles or molecules from within them into the extracellular fluid surrounding them. And then finally, we talked about the structure of ATP and how ATP works at a molecular level to either store energy or release energy when it is needed by cells for cellular processes. As usual, it's often a good idea to repeat things over and over again in order for them to be more likely to stick in your mind. So I would suggest that you rewatch this video a few times And each time you try to fill in the worksheet by writing the material down. Eventually, you should then start trying to test yourself by seeing if you can refill the worksheet without looking at the video and without looking at previous notes or copies of the worksheet. Eventually, once you get to a point where you can refill it from memory, then you can feel quite confident that you have retained the material. It's also helpful to do past questions on these topics so that you can familiarize yourself with how questions are asked in exams. If you found this useful, then please click the like button at the bottom and subscribe to get notified whenever I put out new videos. I hope you found that useful, and do come back and watch more videos in the future. Thank you.